Welcome back! Today we will be talking about developmental networks. So I was talking to Hagrid recently, and he was giving me some updates on Norberta. Yes, Norberta was a girl. So he was telling me that Charlie Weasley, this is my accurate portrayal of Charlie Weasley, has been looking after Norberta, and he has hypothesized a developmental network that allows for spikes to show up on her skin, but not on her wings, head, or belly. Now here is the developmental network that Charlie Weasley sent over. Now before we start, the arrow represents that the gene is being activated and that it will be produced. The T-bar represents that the gene will be repressed and it will not be produced. Now, since Charlie doesn't know too much more about genetics, we're gonna analyze some cells that he gave us and we're gonna see if they align with the phenotypes. So we're gonna start with the wild type first, meaning that there are no mutations in the network. So let's start with S. We know that S is present, and if S is gonna activate P, that means that P is gonna be present. Now, since P acts to repress I will not be present. When I is usually present, it represses K, but since it's not present, K will be produced. And since K promotes E, and E promotes spikes, which means spikes will be present. Our next genotype is a mutation on the P gene, which means P will not be produced. Now, if P is not produced, it is not able to repress I, which means I will be present. And since I is present, it is able to repress K, and K will not be present. Since there's no K to activate E, E will also not be present, which means that the spikes will be absent. Type is going to be a mutation on the I gene, so I will not be produced. Since there's no I to repress K, K will be present. And since K promotes E, E will also be present, which means that spikes will be present. So our next genotype, we have I and E both not present. Now our main focus will be on E because I is upstream and doesn't have as much effect as E does. Now, since E is just not present in general, we know that the spikes will be absent because E has to be present in order for spikes to appear. For our last genotype, S and K are not going to be present. Now again, we don't need to focus on S because it's more upstream. Now since K is not present, it will not be able to activate E. And now because E is not present again, spikes will be absent. We saw that Norberta didn't have spikes on certain parts of her body. She lacked spikes on her wings, belly, and head. So why wouldn't all the skin cells produce spikes if they have the same genes? Well, we learned a form of transcriptional regulation in a previous lesson that can repress the transcription of certain genes, DNA methylation. So we talked about this recognition sequence called CPG, and that is frequently seen before the start of transcription sites. When this sequence is heavily methylated, as seen on the cytosine to the right, here is the methyl group, then transcription will be reduced. So we could expect to see heavily methylated cytosines in the cells of the understomach, of the wings, and on the head. Now that is the end of the lesson. I hope you guys do well on your OWLs.